We are the Keratins, and this is our video about the process of the cell cycle. Enjoy! The cell cycle is preparation for division and the division of cells. It is a continual process divided into these three main stages: interphase, mitosis, and cytokinesis. After this cycle is finished, two new daughter cells are produced. The first part of interphase, which is the first stage of the cell cycle, is G1 phase. During G1 phase, the cell grows and produces proteins, lipids, and carbohydrates. It also replicates organelles during this phase. Also during G1 phase, as shown to the right, mRNA synthesis begins. If there is any type of mistake in the cell cycle, the cell is not going to divide, but instead it will stay in G0 phase. Because the cell is not going to divide, there is no replication of DNA in this phase. Because the cells that stay in G0 phase have problems or mistakes, apoptosis, otherwise known as controlled cell death, may occur. As shown to the right, the DNA is replicated during S phase. During S phase, the chromatin is preparing to become chromosomes. In G2 phase, the DNA gets checked for errors after it's been replicated during S phase. These errors are checked for at proteins called checkpoints, which ensure that mitotic events occur in the correct sequence. Now that the cell is done preparing for cell division, it can finally undergo cell division. There are two parts of cell division, the first being mitosis, which is division of the nucleus. During prophase, the first stage of mitosis, the chromatin forms into individual sister chromatids, which then condense to form chromosomes. As seen in the diagram to the right, each sister chromatid has been replicated in order to form chromosomes that contain two sister chromatids each. Some other things that happen during prophase are that the nuclear envelope along with the nucleolus break down. Also during this stage, centrioles begin to form and move to opposite ends of the cell. As you can see, the centrioles are making spindle fibers, which are very important in the next step. In metaphase, the nucleus disappears completely and the chromosomes line up along the cell's equator. Spindle fibers, which are made up of microtubules, come from the centrioles and then attach to the centromeres of the chromosomes. During anaphase, the spindle fibers retract and the centromeres divide. The chromatids separate and become independent chromosomes. They move to opposite ends of the cell. During telophase, the very last stage of mitosis, the chromosomes uncoil into what we see as chromatin. The diagram to the right depicts how the chromosomes actually look. The chromosomes have split into individual sister chromatids. Also during telophase, the spindle fibers disassemble, the nuclear envelopes reform around each set of chromosomes, and the cellular membrane begins to pinch. Cytokinesis, the third and last stage of the cell cycle, is division of the cytoplasm. In cytokinesis, the chromosomes are uncoiled and form into chromatin. The cell then divides into two cells. Each cell has a nucleus and organelles. Finally, the two new daughter cells can begin the cell cycle. The type of cell that we used to describe the cell cycle was an animal cell. For a plant cell, the process would have been the same up to cytokinesis. During cytokinesis in a plant cell, a cell plate forms across the middle of the cell and then gradually splits the cell into two new cells. this diagram, the nuclear envelopes are reforming around each set of chromosomes. Also, the cell plate is forming across the middle of the cell. Nuclear envelopes are still forming, but the cell plate has grown into a cell wall, separating the two new cells. The two nuclei are now complete. And just like in an animal cell, the chromosomes have become chromatin again, ready to start the cell cycle. And that is our video about the process of the cell cycle. Thanks for watching.